All right, everyone. We are going to get started with the afternoon session. It is my pleasure to welcome Erin from Glencoe, who's going to be telling us about their software that will maybe solve a lot of the problems that we're all facing here. So, Erin, thank you for coming and looking forward to your talk.
Thank you so much. Thanks very much for having us. And yeah, hopefully we can live up to that promise. So hi, everyone. I'm Erin Diel. I'm Glencoe Software's head of product. We've seen a lot of really beautiful images today throughout the talks. And so I wanted to spend a little bit of time talking about image data management and the solutions that we build at Glencoe Software. So I'm going to start by telling you a little bit about our company and what we do. Then I'll describe our core data management platform, which is called OMERO Plus. Then I'll tell you some quick vignettes on solutions that we've built on top of this core data management platform. And then I'd like to finish by showing you briefly some sample data sets that we've loaded into a demo system that we'll have up and running throughout the meeting. And so if anyone has any particular questions or wants to see the system in action, I'd be happy to take you through that. Okay, so let me start my timer. Great. Okay, so at Glencoe Software, we aim to build the most comprehensive scientific image data management solutions at a global scale. And that's really dictated by the customers that we work with, who are often global entities. And one thing that's really important to us is that our solutions are backed by open source technology. We think this works really nicely because it means that our technology is constantly evolving to meet the emerging needs. I think we've seen that especially spatial biology is a very fast-paced environment to work in. But we also work with some of the world's largest organizations that need a lot of security and stability in these solutions. And so we like to pair this open source software with industry-leading service and support. And fundamentally, we think data management is really interesting because we're aiming to provide you faster and easier access to your data. We know these data sets are very complex. And so if you can access your data more easily, that enables you to make more insights, more discoveries, which is ultimately what you're trying to do with your data. So we do have an official basis in the UK and in the US, but our team is fully remote. We've actually been fully remote since we were founded. And I'm based in Boston. I have a colleague, Mark Bruce, who's here with me, who's here in California. And our team is distributed across the US and Europe. Collectively, we have many decades of experience in the life sciences and imaging because we work with a lot of people doing life sciences and imaging. We also do a lot of work in data publishing. And of course, we're a software company, so we have a very strong engineering team. The customers who we work with are pharmaceutical companies, large academic institutions, and generally people working in bioimaging research and also publishing. So we're a small company. There's 18 of us at Glencoe Software, but we are partnered with this group called the Open Microscopy Environment, and there are over 30 contributors to that project. And in fact, Glencoe was founded back in 2005 by two of the creators of OME. So if you're not familiar with the Open Microscopy Environment, I'll tell you a little bit more about them as we go through the presentation. But this is a grant-funded, community-driven group of people, and we are their exclusive commercial partner. And so a lot of the solutions that I'll tell you about today in terms of the core data management solutions are available open source. And so you can try them for yourself today for free if you don't want to enjoy the sunshine instead. And so why might any group work with Glencoe Software? Why might there need to be a commercial partner of the Open Microscopy Environment? So the first is that we offer commercial licenses for a lot of these products. This is important for some of the entities that we work with. So these commercial licenses provide various warranties and indemnification that are important for some of the world's largest pharmaceutical companies or research institutions in general. We also customize the data management solutions that we deploy for our customers. So sometimes this is just at the level of we are deploying something on your infrastructure and we're working with your data. And so we need to make sure that we don't create a one-size-fits-all solution, but we create an architecture that makes sense for the kinds of data that you use and the way you're using that data. But it could also be at the level of writing a custom script or a custom integration with, say, an image analysis platform or a LIMS system or any other thing that you're using to work with your image data and other kinds of data sets. And the third piece is probably most important to our team, and that's our service and support. So we develop really hands-on and long-term relationships with our customers. Some of them we're meeting with on a weekly basis. We have customers who we've worked with for years. And so we really like to understand the evolution of how they're using imaging data and make sure that our products and our people are supporting them as best as possible. So we think collectively these two groups, the open microscopy environment and Glencoe Software, have really built the standards for how we read bioimaging data and how we manage bioimaging data. And so the two products that you might have worked with on the open source side are called Bioformats, an image translation library, and OMERO, a data management system. And the commercial equivalents of these two products coming from Glencoe Software are Glencoe's Bioformats and OMERO Plus. And so these are those two products that would have a commercial license associated with them. And the two on the left, Bioformats and OMERO, are the things that you can use open source today. Now, Glencoe Software not only develops and supports our commercial products, but we also do a lot of development that we bring back into the open source community. And so this is a real partnership, and it's incredibly important to us, not just our customers with commercial licenses, but members of the open source community as well. So just a couple of numbers and statements to back up this statement that we've become the sort of standard for bioimaging data. So Bioformats is an image translation library, supports over 150 different file formats. And so if you think about the various vendors and subdomains within bioimaging, we have pretty good coverage of the various kinds of imaging data that you might be producing. And there are a number of different applications that use bioformats to read from a variety of different proprietary file formats. And we have a little bit of information on when Bioformats as a library is launched in these applications. So these are things like Fiji or Cell Profiler that use Bioformats. And actually, Bioformats is launched over 100,000 times per day for people trying to read their bioimaging data. And so some of those numbers are why we really feel that even from the open source community, these have really become sort of standards for bioimaging. I mentioned earlier that OMERO Plus, as a commercial product, offers full warranty and indemnification, and we're trusted by global organizations, some of the top 10 pharmaceutical companies in the world, and some of the largest universities in the world. Now, I'm going to talk a lot in my later slides about spatial biology, because that's what we're here to talk about. But I want to emphasize that we work with customers across various imaging domains. And so it's not exclusively whole slide images and digital pathology that we work with at Glencoe Software. In fact, some of these tools, both from Glencoe and the open microscopy environment, were sort of formed with electron and fluorescence microscopy, sort of basic methods of producing imaging data that have been around far longer than some of the really exciting techniques that we've been talking about today. We as a company at Glencoe Software end up working a lot with digital pathology and high content screening. This is largely based on the fact that pharmaceutical companies are doing a lot of HCS and digital pathology. We also do a lot of work with medical imaging. I mentioned earlier that we work in publishing. This is in part the images and videos that you see in line when you're reading a data publication in the browser. That's some work that we do with a variety of publishers. But we also work with our customers to use the data management platforms that we develop to actually publish their data if they need to for a grant reason or just because they want to make that data public. And there are others as well. So if the images that I showed today or the solutions that I showed today don't speak to a particular imaging use case you have, we'd be happy to talk more about it. We may already be working with that kind of imaging data, or we may be able to add support for it. So before I dive into all the details of what OMERO Plus and Bioformats really are and what they allow you to do, I want to step back and talk about why imaging data is so fundamentally hard to work with. I know probably most people in the audience are working with imaging data in some form, but some of you might be coming from more of the genomic space, and maybe you haven't worked with imaging data as extensively previously. So I want to talk about why this kind of data can be really fundamentally challenging to work with. So the first is that it's often quite large. And so on the right there, you can see that this image, which is a pretty standard whole slide image, is tens of thousands of pixels in the X and Y dimensions. Now you might have 3D or 4D data sets that are even larger than just in the XY dimension. But we know that we often aren't trying to read all of this data at once. We're trying to work with it piecemeal so that we can actually handle all of that data. It's maybe a terabyte size data set in total. There's also a lot of complexity in terms of the formats that this data is written in. And so we have a lot of fabulous vendors in bioimaging who are motivated to write data off their instruments as quickly as possible. And so they create file formats that enable them to do that. And that's not fundamentally a bad motivation, but what it leaves us with is a lot of proprietary file formats that are not interoperable. I can't open vendor one's format in vendor two's software. So it becomes really challenging to have sort of cross-modality research and analysis or even visualization of this data. And so here's just one example of this particular image in the SVS format. And so you might have imaging data from across different vendors, and it's all in a different format, and all of those formats are proprietary. This image data also doesn't exist in isolation. It often has metadata associated with it. And so just having the pixels themselves probably isn't very useful to your research. You need to know what this image is. And some of that information might be historical. And so it's particularly important to keep track of this information over time. And so on the right there, I'm highlighting some clinical metadata. This is an image from the Cancer Genome Atlas. So that might be one kind of metadata that you care about. Another kind of metadata that's highlighted in the center of the slide is a spatial annotation on the slide. So another kind of metadata, completely different than a text-based annotation, but again, something that means that this image does not exist in isolation. And the images aren't in isolation from one another. I would venture to guess that your average data set is not a single image. It's often many images. And so when you think about a visualization workflow or analysis workflow, you have to not only contend with the size of a single image, but the size of the entire collection of images that you have together. And it might be relevant to sort of keep track of these as a collection over time. And so again, having images be complex in and of themselves, but then have them not exist in isolation just sort of compounds that complexity. We have another fabulous advantage, sorry for the knocking my microphone. We have another fabulous advantage in the bioimaging field, which is that we have lots of fabulous analysis tools that are constantly evolving, which is great, but it also presents a challenge. I think a lot of the grad students and postdocs in the room would have experienced this, that every day you feel like you have to learn a new tool to analyze your data in the best possible way. And so this is a great advantage, but it is also fundamentally a challenge. And so as you're trying to keep up with the newest tools, this is just a selection of a few, and I'm highlighting them purposely because these are things that we have built integrations with. And so you want to be able to not only access your data, but be able to use them in an interoperable way between these different solutions, because you're often not just going to pick one and stick with it for many years to come. Okay, so let's talk about this core data management platform called OMERO Plus and why it might help address some of these problems. And so fundamentally, as a data management platform, we're enabling you to view, share, annotate, and analyze your very large image files from anywhere in the world from a single software interface, okay? And so the two components that are really important here are actually not just OMERO Plus, but also this image translation library called bioformats. So as I mentioned briefly earlier, we think this has become the global standard for both image data and metadata. And because it's a translation library, what that means is that it allows you to read from these proprietary file formats without requiring that you convert them. So it's translating in real time from these various file formats. It's utilized by OMERO Plus to allow you to access both the image data and the metadata. It's compatible with imaging data from a number of different imaging domains, which means that in a system that's using bioformats, you can take imaging data from a variety of different platforms and do the same things with them, whether that's looking at your images, analyzing your images, annotating your images, because you're using bioformats to make all the data effectively look the same, to translate between those different formats. Importantly, it's accessing the metadata as well so that you don't lose the context of where that imaging data came from. And there are a lot of sort of industry standard tools that are using bioformats already. So a few are listed there: ImageJ, MATLAB, Amara, Cell Profiler, and there are many others. And I mentioned earlier, we support over 160 different file formats currently. So OMERO Plus is using bioformats to allow you to read from these variety of different proprietary file formats. So this data management platform now enables you to view your data from anywhere, even if your data is far away from you. You can manage your scientific images and their associated metadata. That maybe allows you to share your data with your colleagues, but in a secure way, so I can control the fact that I am sharing it with someone, but also what are they allowed to do with that data when I share it with them. You can analyze your data, so accessing bits of data at a time so that you can, say, segment the image. I'll show you some examples of that in a moment. And you can publish your data. This might simply be the reality that I can find the images that I care about so I can publish these data sets, but it also might literally mean that you're using this data management platform to make data public so you can send someone a URL to have them take a look at your imaging data. So that would go hand in hand with the concept of sharing, but taking it to the extreme of sharing it publicly. Okay, so let's think a little bit about how this platform looks like to you, the end user. So OMERO Plus is something that's running on a server somewhere in a data center in your university or maybe in the cloud. It actually doesn't really matter. And of course, it has some imaging data that it's aware of. And so these might be the original files that came off the microscope. Remember, we don't have to convert them because we're using bioformats. We have some search indexes that allow you to find the images that you care about. So maybe you're searching by something in the file name or some metadata that you have. But ultimately, if you have the data under management, you might want to be able to search for it. And of course, you have some metadata. Now, some of that metadata is coming from the original file itself, some acquisitional metadata, but it might also be additional metadata that you've added after the fact. So this is in particular clinical metadata or experimental metadata that isn't always in that original file. It's written down in your lab notebook somewhere. So you might actually put it into this OMERO Plus platform in order to associate it with the image data. And of course, it could be very complex metadata, which we often refer to as analytics. And so it might be very rich tabular data that you have representing some analysis, some segmentation, some genomics data. And so that kind of analytical data can also be associated with your images. But you are not sitting by the server. Some of you might be, but you don't have to be. You're on the other side of the internet accessing your images. So you might be on the acquisition system and you might be actually importing your data from the acquisition system into this data management platform. You could be interacting with any number of our clients that we have. There are some open source tools that you can use to interact with data in OMERO. We've developed some interfaces as well. And one thing that I want to highlight, just very quickly here, is that this is very pluggable. And so you might be using any number of the clients that I'll tell you about today, which are things developed by Glencoe Software. You might use open source tools that have been developed by the open microscopy environment to interact with your imaging data in OMERO, or you could build your own client to interact with your imaging data in the way that matters to you. So we saw some great examples from people like Niels this morning on interacting with data in the browser. And data that's under management in OMERO is very purposefully open to you. So the APIs that you can use to access your image data, or even say, hey, where's the file so that I can go interact with the file directly. We enable that because fundamentally managing your data should not happen in a closed system, because then you're not really solving the problem of easy data access that you want in a data management platform. And then the last component there is processing. And so there are some ways in which you're interacting with your data, which are actually interactive. You're sitting in a browser on your laptop looking at an image, but you might also think of a script, for example, interacting with your imaging data. So pulling some pixels, pulling some metadata, running some segmentation. And so the client that you develop could actually be a Python script, for example. And again, all those APIs are completely open, regardless of whether you're using open source OMERO or the commercial version OMERO Plus. So interoperability is a term that we use a lot. This is a really important concept in the data management solutions that we build. And these are just a few examples of the tools that our customers are using very frequently to interact with their imaging data in OMERO. So some popular tools that might be familiar. So open source tools like QPath or Fiji or Cell Profiler for image analysis. You might also be using commercial tools like Visiopharm or Halo. We've built connectors for those platforms. Or you might write your own scripts. So you might be using MATLAB or Python, and we have APIs to allow you to access your image data and metadata in exactly the ways you want, and then use all your preferred tools to analyze or visualize whatever you need to do with your images. Okay, great. So now I'm going to transition into talking about some of the interfaces that Glencoe Software specifically has built on top of this core data management platform called OMERO Plus. So remember, if these interfaces don't speak to you, you can build your own, or you can use any that are out there. I'm not going to describe all of them today, but I'm just going to describe a couple that I think speak to some of the spatial biology challenges that we've worked with our customers on. And we have this demo system set up. So if you have other use cases, happy to talk about those more specifically. So the first one that I want to talk about is PathViewer and PathViewer Grid. So this is a browser-based visualization tool that allows you to view your images, analyze your images, and annotate your images. And it is specifically tailored for the digital pathology workflow. So if you've ever worked with OMERO before, you might know that it aims to be as platform agnostic or domain agnostic as possible. So it's meant to be very open, interoperable. You can put whatever kind of image data you want into it. But when you're doing a pathology review workflow, there are some specific assumptions that you have about the kinds of imaging data that you're working with and what you're doing with those images. And so we wanted to build an intuitive and clean interface that made sense for that digital pathology type workflow that really put sort of the slide, the image at the focus, and had a lot of easy annotation tools that you could work with. And again, this is in the browser. And so you don't have to install anything additional in order to look at your images and annotate your images. So we have things like freehand annotations, drawings, and measurement. You can rotate, flip, and sort of zoom in and out on your image, all the things that you'd want in an image viewer. And because it is sort of built with these whole slide images in mind, we also have the concept of a slide preview or that overview image and a barcode image. So all of that metadata, which in that case is actually an image that represents metadata, that information is kind of front and center for the pathologist who's reviewing. You can draw region of interest annotations in PathViewer, and you can add metadata to those region of interest annotations. And so this speaks to kind of the hierarchy of metadata in OMERO. So I really highlighted that images don't exist in isolation. And I want to emphasize that the metadata that you care about, there's lots of different places where that metadata might make sense to exist. So it could be image level metadata. It could be metadata that makes sense at the collection of images, or at a region of interest annotation that you've drawn where you want to add some information about specifically this region of the tissue. And so you can add that metadata at that level within OMERO and specifically within PathViewer as well. And there's all sorts of advanced viewing options to change the rendering settings of your image. And so I want to highlight in particular these highly multiplex data sets that we've seen some examples of today, where you're not looking at a standard bright field H&E image, but your image has tens or even hundreds of channels on it. And that is totally valid within OMERO and within PathViewer. And we've added some little tools that try and make this a little bit easier to work with in terms of the complexity of those multi-channel images. And so what you see there on the left is the concept of channel groupings within PathViewer, where you can organize groups of channels together, maybe based on a round of a cyclic protocol, or maybe based on some gene ontology information. So it could be more experimentally or more biologically meaningful groups of channels. You can create multiple different groupings. You can duplicate channels across different groups. And so you create these conceptually meaningful organizations to your channels instead of having a massive 100 channel image that you have to then click through on off a variety of different channels. And that brings me to another interface, which is called PathViewer Grid, where you can compare multiple images side by side. Now, PathViewer Grid, it's actually multiple mini PathViewers in one. And so all the features that I described previously in terms of viewing your images, zooming in and out, annotating your images, possible here. But now you have multiple images in view. You can synchronize across those views, zoom in and out, but also annotate them at the same time. And importantly, for these highly multiplex data sets, you can do the same thing within an image. And so let's say you have 10 different groups of channels and you want to view those groupings of channels side by side, even within a single image. So whether you have serial sections, highly multiplex data, or the same section that you've imaged in multiple modalities, you can view them side by side in this application. And again, you have all the same features that were originally available in PathViewer, but now in this side by side view. One other extension that we've recently made in PathViewer is to allow you to explore analytical results overlaid with the original image. So again, this is all in the browser. And you can now not only look at the images themselves. So here we have a nice fluorescence image with 3 channels shown, but this is actually a highly multiplex data set with, I think, 20 channels on it. And here overlaid now in those white outlines is a nuclear segmentation that was performed. And so you can view that again in the browser overlaid with the original image. But we also have some analytical data associated with each of those objects. And so in spatial data, I think we've had a few people highlight that you do something like a cell or nuclear segmentation, you're talking about potentially millions of objects in your image. And so this is an example. In this particular image, we had about 1.5 million objects on this particular slide, and you're seeing those overlaid with the original image. And we have about 100 or so measurements that we've taken of each of those 1.5 million objects. And here, I'm just color coding the objects based on those measurements. And so in this case, we color coded it based on the circular compactness of the object. And so you have some nice round objects in yellow and some more elongated objects in blue. And then everything that didn't fall within the tails of that distribution is not colored in, not filled in. And so this is just a simple example of how you could take those very rich analytical results, enrich not only in the sheer volume of results, but also the complexity in terms of multiple objects and multiple measurements of each of those objects, and do a little bit of data mining and data visualization here within PathViewer. But how would you produce those results in the first place? So I highlighted some integrations in earlier slides where you might write your own custom Python script, for example, to segment your images, or you might use one of these commercial platforms like Visiopharm or QuPath, being an open source option. But we recognize that not only did our customers want to look at their images and these analytical results within the browser, but they wanted to be able to kick off some analysis in an equally intuitive interface, like opening up a web browser and hitting go on your segmentation routine. And so we built the OMERO segmentation connector to allow for that scalable, but still interoperable and customizable image analysis from the browser. So just to frame the problem that we wanted to solve, so we want to take this image on the left and we want to get the image on the right, which is, in this case, a nuclear segmentation. You can imagine your preferred segmentation, but here it was the instance-based nuclear segmentation. And the challenge is that the image on the left is massive, like we described earlier. So, we've got, let's say, 100K by 100K pixels. We're not going to load the entire thing into memory and run our segmentation because not only do we have one image of that size, but we have 500 images of that size. So how do we run this except on a very beefy workstation that we book for a week of time in order to run that segmentation? Well, what we want to do is we want to divide this image up into tiles, more manageable chunks of data, and then run our analysis on those chunks of data and then pull everything back together. And so that's exactly what we built with the OMERO segmentation connector, which takes your original images, allows you to divide them up into tiles, run your per tile segmentation, then pull everything back together, which if you've ever done this is trickier than it sounds, right? Because you want to have some overlap between those tiles and make sure you don't miss objects on the tile boundaries. But then you're left with, let's say, millions of objects across your image. And then you want to measure a bunch of interesting features about each of those objects that you can do some post-processing with downstream. And importantly, all of this is done via this central data management platform of OMERO Plus, which means that not only are you kicking off your analysis from an intuitive interface, such as within a web browser, but you're also bringing all the results automatically back into OMERO. So the results of your analysis are managed alongside the images. This not only enables workflows like the one in PathViewer that I showed earlier, we could look at those results, but it also means that you can share your results with your colleagues a little bit more easily. You just have to send them a URL instead of sending them many gigs of CSV data. And it also means that you can come back to that analysis potentially years later and know the parameters of what analysis was run. So it makes it much more reproducible, where you can track not only the images, but the analysis that you performed on those images. We've also seen ZAR in some of the other presentations. So that's one of the technologies that we're using to make this tiling of the images a little bit easier so that we can access subsets of the data at once, not only subsets in terms of spatial tiles, but also multi-resolution data. I'm not going to go into too much detail, but for those who are interested in that particular topic, happy to discuss it in more detail. One thing that's really important to us is that we don't want to prescribe what analysis you're actually performing. And so I had the nebulous statement in the previous slide of per tile AI segmentation. And we promise we're not trying to reinvent how you segment your images. We're actually aiming to use we're aiming to support the most popular AI segmentation algorithms, many of which are open source and we can use in this segmentation connector. And so some examples of that are things like Stardist and CellPose. For more classic computer vision approaches, and especially in the high content screening community, they're using a lot of Cell Profiler. And we've recently added support for a format for AI models called Onyx, which isn't necessarily an AI method in and of itself. It's just a format that you use to describe your models. And we're hoping that that opens up a lot of possibilities for our customers who are really interested in developing their own AI models and just want to plug them into this segmentation connector. It's also very customizable from the perspective of what compute you're actually using. And so if you want to use some cloud compute resources or you have an on-prem HPC cluster that you want to use, we try and make this very flexible. Or if you say, no, we have this dedicated machine, we just want to run all our segmentation there, we can do that as well. And I mentioned earlier that not only do you segment the images, but we calculate a number of features about each of those objects. So per object intensity, morphometric, and spatial features are things that are calculated automatically. But you actually get the geometries back out of this pipeline. And so if you want to take further measurements of those objects, you have full access. Again, there are APIs to access the output data from such a workflow. So I really want to emphasize that we're working with our customers to understand how they are analyzing their images and what, for example, segmentation methods they're using, but we're not trying to reinvent image analysis. We're not trying to say we're analyzing the data better than anyone else. We're trying to provide an interface that allows you to execute image analysis at scale and nicely from within what we think is a pretty intuitive interface of the web browser. And importantly, those analysis results and the conditions of that analysis are managed alongside the images. And so it makes things much more traceable and reproducible over time. So let's think about how we actually look at these image analysis results. So I showed you some examples earlier from PathViewer. We had a nice nuclear segmentation overlaid with the original image. And we found that we had a problem with these very large images, millions of objects. It was fine to visualize those objects, but when you're all the way zoomed out on this piece of tissue, there's some interesting spatial patterns that we can already appreciate with this image. We don't have to zoom all the way in to see where there are some interesting patterns in the tissue. But if I try and overlay the segmentation results, it at best looks like there's some sprinkles on the image and you can't possibly appreciate the nuclear segmentation that you just spent lots of time performing. Now, of course, you can zoom in and now I can see, all right, there are nicely segmented nuclei here. But what about when I want to appreciate the patterns in the tissue at this level? I want to know, okay, from a high level, where are the hot and cold spots on the image that I want to look at a little bit more closely to QC my analysis results? And so we are using fundamentally what we're overlaying with the image is a label image. And everything that I've shown you so far is the actual segmentation result that's overlaid with the image. But the concept of a label image is flexible. It doesn't have to be the actual objects that you segmented. And so with some of our customers, we created more of a heat map style view of these objects where we now tile across the image with these hexagons. And each of those hexagons represents an average, a local average of the objects that are sitting underneath that hexagon. And so here you can see, for example, I'm no longer color coding the individual objects based on some measurement, but I'm color coding this arguably meaningless hexagon with, okay, in this area of the image, what is the average, for example, area of the underlying objects? And so if I actually zoom in, I can say, oh, okay, there's an interesting hot or cold spot in this part of the image. And what you can see here is that from blue to brown, we're color coding based on the local average of the area of the underlying objects. And so you can say, oh, I want to go find that hotspot of really big objects. And then, of course, once you zoom in, you can switch back to the actual nuclear segmentation and just color code the primary objects themselves without having to look at this heat map style view. And so we just created this sort of parallel representation of the segmentation results so that people could explore at different scales of their original images. And then you might take those results And you might say, okay, I have previously a more continuous view of blue to brown, what were the size of the objects. But now I just want to create a classification. I want to find my big objects and make them magenta, and I want to find my small objects and make them yellow. And now I have two classes of objects that I'm looking at on my image. And this works very nicely when you have large numbers of objects that are classified as big or small. But you might be looking at rare populations within your image. It might not be as simple as looking for the big and small objects. And so what happens if across this very large image, you've mined through all these interesting results and you've found the objects that you care about, the class of cells that you care about, and there are 5 of them across the entire image. And how do you find them? You have to scroll through, zoom in and out, and find these teeny tiny objects that, remember, are really hard to see at this scale where you're all the way zoomed out. And so we recognize that while the tissue context is really important, when you're looking for rare populations, in a view like Path Viewer, you're trapped within the tissue context. You can't say, okay, you found 5 cells. Can I just look at the 5 cells side by side to see if maybe 2 of them aren't actually what I would consider, say, a macrophage or whatever I'm looking for within this tissue? And so we built another interface called Pageant. Again, this is in the browser, but it's also similar to Path Viewer, meant to allow you to interactively mine through your data. But this is specifically allowing you to balance those object level results and the tissue context that is still sometimes relevant. So the idea here is that within an image, you have a number of objects that you found across the image. And while you do want to appreciate where a given object came from, you also want to be able to have an object-centered view. So in this case, these are nuclei, and I want a nice grid of all the objects that I found within my image so that I can QC them very quickly or mine through them very quickly. And so this was the intention with which we built Pageant. And so here, what you see is synchronized views between these 3 different panels here. The top is the actual tabular data, so a row per each of your objects. On the bottom left, you have your object thumbnails where you can see each of these objects side by side. And then on the right, you see the actual image. And these 3 are synchronized. And so you can click through individual objects, you can click through individual rows of your data table, and it will bring you to the right location, for example, in the image. And so you can still see the objects themselves centered in the view, see them side by side, look for interesting outliers, and then say, let's go see if that was in a weird part of the tissue. Maybe it's on the squashed edge of the slide, and so we don't need to worry about that particular outlier. Similarly, you can use the data that you have on each of these objects to filter and mine through the objects that you care about. And so here, just a simple histogram visualization for a particular feature that you calculated for all your objects. And you can find whether you want, as my previous example, the smallest objects, filter them out. And I think actually this is filtering for the more elongated or more circular compact objects based on a selection of a subset of the histogram. And so you can take those millions of objects and actually slowly mine down to the few that you might really care about. Okay, so with that, I want to just quickly show you, I mentioned that we had this demo system set up with some relevant spatial biology data sets loaded into it. I'm not going to take you through many tens of minutes of demonstration of the platform, but I just wanted to highlight what we have loaded in. So if any of you are particularly interested in seeing things in action, we can go through it either today after this talk, or we're also here tomorrow and we can talk more extensively then. So I'm going to end this and I'm going to switch to my browser here. Okay, so we have a demo system set up with some public data sets from a lot of the popular vendors, some of which we've seen throughout presentations today. And so I'm just going to quickly click through some kinds of data that we have and just some quick highlights of what we could sort of go through together in a live session if anyone is interested. So here I have some MIBI data sets from IonPath. And so this is a great example of how you can use your channel groups to organize highly multiplex data. So this has something like 40 channels. And so we've organized those channels into groups so that you can intuitively turn on and off groups of channels at once. And you can also see, okay, that marker corresponds to something, immune related, for example. And then I can use PathViewer Grid to look at, this is a single image, but I'm looking at 4 panels that are each of those channel groups that were on the previous screen. And so I can navigate this highly multiplex data. I can see sort of all the channels at once in a way that my visual system can accommodate by viewing some subsets of the channels side by side. I have some data from NanoString and Gold Star to NanoString. They write an OME TIFF, which is one of these open standards for file formats. And they also have really lovely mask and other ROI data in that OME TIFF itself. And so if you import this directly into OMERO, we didn't do any post-processing of or pre-processing of this data. And you can see you have an image with a mask already overlaid. So quite nice that they already write their data in this open standard that's sort of easily ingestible by the platform. So you can see those orange and green masks there from the, this is geomics data. I have some Visium data as well. So here I wanted to highlight that in a lot of my label image views that we showed during the slides, a lot of it was nuclear segmentation data, but a label image can really be anything. And so here it's the spots from your Visium experiments. And so you can see that these are just nicely regularly spaced dots covering the image. And here I'm color coding them based on the cluster that they were assigned. I think there's 10 clusters across all of these spots. And so you can take some of that data that's been post-processed out of the Visium platform and overlay it with the imaging results in PathViewer. And similarly, you could explore these kinds of results in Pageant. So even if you're talking about spots in the bottom left there, you might want to, this, for example, is looking at all of the spots that were associated with cluster 3. And now I can view them side by side. I don't have to search through the tissue for all the spots that were identified as cluster 3. But if I want to click and say, okay, let's zoom to that spot on the image, I can do exactly that. So these are just a few of the sample data sets we have. We have some of the MC micro data sets. We have some from the RareSite Orion system. And I already mentioned some of the others. If there are others that you're particularly interested in, we can try and load them up during the meeting. So happy to take any questions and also happy to go through some of the sample data sets that we have live with anyone interested. That's it. Any questions?
Quick question.
Yeah.
What happens to the ownership of the files when you upload it into your server in the sky?
So that's really important. The OMERO Plus solution or the open source version OMERO is running on your infrastructure. So this is not a software as a service product. This is something that we deploy in your data center at your institution, and you're using data on your storage. And so it's yours. The ownership never changes. And in fact, nothing is even running on Glencoe Software resources, even in the commercial solution.
But you say it's running on the internet, no?
So it's running, the OMERO Plus application is running on a server, and it would be running on a server in your data center, or if you're using a cloud provider, it could be running in your AWS account, for example. But it's infrastructure that you own. And so the data is sitting, just like you might use some network attached storage now to store your microscopy data, it would very likely stay there, and you would have the OMERO Plus application running on a server, and you'd be importing the data into the system, and it would stay on that storage. And then you as an end user are opening up a web browser to look at your image data, but it's still sitting on that institutional storage.
Okay, thank you.
Yeah, of course.
It's a very cool talk. So I saw you've got this deep focus on reproducibility and being able to go through and look at the steps of what happened to the data, but you also have a lot of pluggable outside systems in which you can do the manipulations. So do you have some way to track the provenance of that? So if you run like a Python script, can you give a copy of the Python script somewhere that can be embedded that you can go back and revisit? Is something like that possible or within the scope?
Yeah, absolutely. So a couple of options there. I mean, fundamentally, if I use the API in a Jupyter notebook to analyze my imaging data, it's not by default that there's not an audit log of every single action that the user took. Now, there is actually an event log in OMERO that tracks some changes. So there's a little bit of that. But fundamentally, it enables you to say, oh, I ran this Python script. I'm going to bring it into OMERO so that my colleague can run the same Python script, but it doesn't require that you do that. So there is still a little bit of user responsibility.
So you can add in you can add in metadata, you can put in code as well in some way. Okay.
Exactly. And in fact, there's the concept of something called an OMERO script, which I'd be happy to demo if anyone is interested. But it's just a Python script. But there's a way that you can load it into the OMERO web system so that you can click in the browser in order to run that script. And so it's really easy then to, as the data scientist, you write the Python script that you want someone to be able to execute, but they don't have to worry about running a script or setting up their local Python environment. They're just working in the browser and you've sort of set everything up for them. So that's a really nice way to take a Python script and make it available to another user.
Thank you.
Yeah, sure. And that's all sort of core open source OMERO as well. The concept of OMERO scripts and the APIs in general are all part of this open source core. Great. Okay, so happy to show the system live for anyone who's interested or answer any other questions. Like I said, Mark and I are here for the afternoon today. I know that the talks are over, but we'll be hanging around a little bit and we'll be here tomorrow as well.
Okay, awesome. Well, join me in thanking Erin again for a great talk.
Thank you.
Okay, that's it for today. So we will see you guys back here tomorrow for the third day. And they're sticking around, as they mentioned. So if you want to come up and ask private questions, you're welcome to. If not, we'll see you tomorrow.